good afternoon everyone. Once again I welcome you all to this short-term training program of NAHEB CAST. So in continuation with the series of experiment which we are doing under this particular training program. Today we are fortunate to have Dr. Anil Dahuja sir. He is professor at Division of Biochemistry IRI. So now I request sir to please brief about today's experiment and then further we'll take up this experiment in the lab. Sir.
Okay, thank you Dr. Anil. So today basically we are going to do the estimation of a very important enzyme, that is the beta-glucosidase. So I will first brief you about the importance of the beta-glucosidase and why the researchers all over the world, they are so much interested in this particular enzyme. So as you all must be knowing that in case of plants and of course in different kingdoms you have different types of metabolites present. So these metabolites, they could be primary metabolites or the secondary metabolites, and you know the difference between the primary and the secondary metabolites. The primary metabolites, they are required for the growth and development of the cell while or the organism, while the secondary metabolites, they are required for the survival of the organism. And usually you will find that these secondary metabolites, they are present in their conjugated forms. So that means they are always linked with certain moieties. These moieties, most commonly they are the sugar moieties. And what could be the possible reason for this? Why they are present in the conjugated forms, these secondary metabolites? Do you have any idea about it? The first and the foremost reason is that the conjugation, it enhances the solubility and the stability of these secondary metabolites, because when you are adding the sugar moiety, you are adding the actually hydroxyl group to the metabolites, and these hydroxy group, they enable the metabolite to be solubilized in the aqueous environment, and they also enhances the stability of that particular metabolites. So this conjugation, it is good for the plant, because the metabolite, it is getting solubilized and stabilized. But if you see for the human consumption, this is not that good, because it has been found that the bioavailability of these conjugated forms, it is much lesser as compared to the free forms or the non-conjugated form. So I'll take them, because our lab, it is working on the a very important phenolics, that is the isoflavones. So you all must be knowing about the isoflavone. It is a kind of the phenolic which is present mainly in the legumes. And why this molecule, it is in demand these days? The demand of the isoflavones, it is related to its nutritional properties as well as the pharmaceutical properties, because it has been found that it is a very good natural antioxidant, just like the tocopherols or the carotenes we have. So if you want to add antioxidant in the aqueous environment or the hydrophilic environment in the food products where the environment, it is hydrophilic, so then you need to have a antioxidant which is hydrophilic in nature, because tocopherols or the carotenoids, they are mainly the lipophilic molecules. So they can be added in the environment where you have the hydrophobicity richness. So for example, if you want to do the fortification of the oil, so there you will contemplate about adding the tocopherols or the carotenoids. But if you are want to add antioxidant in a hydrophilic environment, then there you require the isoflavones. So as I said, that this conjugation, it is good for the plant, because it is enhancing the solubility and the stability, but for the human beings bioavailability, it gets reduced. And human beings, they are actually dependent on the gut microbiome, because we don't have the enzymes to deconjugate these conjugated forms of the secondary metabolites. So that's why we are dependent on the gut microbiome, because our gut microbiome, it contains the beta-glucosidase enzyme, which can deconjugate these conjugated forms into the free forms. And the enzyme involved in this deconjugation process, it is the beta-glucosidase enzyme. So mostly the linkage between the secondary metabolites and the sugars, it is the beta linkage. So you require the enzymes which can hydrolyze that beta linkage, and that's why the name of the enzyme, it is the beta-glucosidase. So now beta-glucosidase is not only required for the deconjugation of the secondary metabolites, it is also required for many other purposes. For example, the activation of the plant hormones, there also you require the beta-glucosidase enzyme. So the point which I want to convey is that it is required for so many purposes in the plant, and that's why the demand for this beta-glucosidase enzyme. And now it could be a generalized beta-glucosidase, or it could be a specific beta-glucosidase. When I say the generalized beta-glucosidase, that means it can act on any substrate present in the cell. So wherever the glucose moiety it is attached, it will remove that glucose moiety, it will deconjugate it. So that is a generalized beta-glucosidase. But specific beta-glucosidase are those enzymes which are specific for a particular substrate. So if sugar moiety is attached to that substrate, only then it will be released by the beta-glucosidase enzyme. So in our lab, initially we were looking for the generalized beta-glucosidase, but then we found that these generalized beta-glucosidase, they are not hydrolyzing over substrate, that is the isoflavones, the conjugated forms of the isoflavones. So in soybean we have different types of isoflavones present. So we have mainly the three different types. One is diargine, then you have genesetine, and then finally you have the glycetine. So these are the three predominant isoflavones present in the soybean, and they are conjugated by the sugar moiety, and then these sugar moiety in turn can be attached with other additional groups. So these additional groups, they could be acetyl, or it could be manolin group. So sugar moiety, it can be attached with the acetyl group, or it can be attached with the malonyl group. So you have 12 different types of the isoflavones present in the soybean. And as I said, so most of them, they are present in the conjugated form, and these conjugated forms, they have the lesser bioavailability. Why lesser bioavailability, by the way? What could be the reason for the lower bioavailability of the conjugated forms as compared to the free forms? Because, yeah. No, when we say the bioavailability, it mainly is concerned about the absorption in the mucosal cells, the intestinal mucosal cells. So when you are adding the sugar moiety to a particular metabolite, what you are doing is you are actually imparting the hydrophilicity to the molecule. And you know the membranes, they are the hydrophobic. So for a hydrophilic molecule to pass through the hydrophobic membranes, it becomes difficult. So otherwise also you are adding a particular moiety, you are enhancing the molecular weight of that compound. So absorption, it depends upon the molecular weight as well as the nature, whether it is hydrophilic or it is hydrophobic. So when the conjugation takes place, so hydrophilicity gets enhanced, the molecular weight gets enhanced, that's why the absorption in the intestinal mucosal cells, it gets reduced, and hence the bioavailability gets reduced, right? So as I said, the beta-glucosidase enzyme, it is responsible for the deconjugation of the conjugated forms into the free forms, and let's see how we can go for the estimation of these enzymes. For example, the generalized beta-glucosidase, so here we can use any substrate, as I said, and the substrate which is most commonly used, that is the para-nitrophenyl glucoside. So you must have read about this substrate, which is being used for many reactions. And this beta-glucosidase, it acts on it, it removes the glucose moiety, and you are left with the para-nitrophenol, and it is a colored compound. and you can read the absorbance of this colored compound at 405 nanometer. So this is basically the principle for the estimation of the beta-glucosidase. We are using a generalized substrate, that is the para-nitrophenol glucoside. It is acted upon by the beta-glucosidase enzyme. You get the formation of the para-nitrophenol, and that is why you get the increase in the absorbance, because this is a colored compound. So this is for the generalized, and if you want to go for the estimation of a specific, for example, say I want to isolate or the estimate the activity of the beta-glucosidase, which is specific for the genistein. So this glucose moiety, it can be conjugated to the genistein, and then we can use that as the substrate, right. So it is a very simple enzyme assay. You just require the substrate and then the enzyme, and then leave the substrate and the enzyme for incubation at 37 degrees centigrade for 30 minutes, and after the end of the incubation period, you will see the increase in the absorbance at 405. Now, how much para-nitrophenol it has been formed? So how will you come to know about that? So you can prepare a standard curve. So in today's practical, we will be preparing a standard curve using the para-nitrophenol, having concentration ranging from 10 micromolar to 400 micromolar. So this is one way that you go for the preparation of the standard curve. Otherwise, what you can do is, if you know the molar extinction coefficient of the enzyme, so that can also be utilized for the calculation purposes. You can see how many micromoles of the products they have been formed per minute. So how to do those calculations? Do you have any idea? If you have knowledge about the molar extinction coefficient of the enzyme. So how many of you are from biochemistry? So sufficient number. So how molar extinction coefficient can be utilized? So Lambert-Beer's law, A is equal to epsilon Cl, and concentration, if you want to calculate, that will be A by epsilon. So absorbance you can know from the reaction epsilon, it is given in the literature, and from that you can calculate how many micromoles or the moles they are formed. And the concentration, units of the concentration, it will depend on the units of the molar extinction coefficient. So if it is in molar, so these many moles per liter, that will be the concentration of the product formed. But your reaction mixture, it is not 1 liter. It is only sometimes 1 ml, sometimes 3 ml, or if the reactants, they are costlier, so then even you can reduce it to say 100 microliter. So that means you have to see how much product it is formed per the amount of the reaction mixture. So you are not going to calculate per liter, but the amount of the product formed in the reaction mixture. So that will vary from reaction to reaction. So the method which we will be following today is, we will prepare a standard curve using the para-nitrophenol by taking the concentration from 10 micromolar to 400 micromolar. So you will take different concentration. So when you are preparing the standard curve, what you will do is, because for enzymatic reactions, what you are doing is, you are adding substrate and you are adding the enzyme extract. But when you are preparing the standard curve, what you do is, you just add the para-nitrophenol, right? And this para-nitrophenol, and then you can see what is the absorbance coming. So you have a standard curve formed, and then in your enzymatic reaction, say this is the OD and this is the substrate concentration taken, or you can say the para-nitrophenol. So you have the OD at 405 nanometer. Now in your reaction mixture, say you have obtained this much OD. So this much OD, it corresponds to this much concentration of the para-nitrophenol. phenol. So, you can calculate how much para-nitrophenol it has been formed in the reaction. Is it clear? So, this is the basic principle, and now we won't be using the purified enzyme here. So, that means we will have to prepare the enzyme extract as well. The sample which we will be using is either, because two samples they have been prepared in our lab. The one is germinating soybean seedlings, or as I said that we human beings, they are dependent on the gut microbiome for the conversion of the conjugated forms into the free forms, and in the gut microbiome we have certain bacteria present, just like the lactobacillus. So, we will be using the lactobacillus acidophilus. So, different strains we have screened the different strains of the lactobacilli, and we have found that acidophilus and rhamnosus, they contain good activity of the beta-glucosidase. So, these bacterium they have been utilized for the preparation of the extract for the estimation of the beta-glucosidase activity. So, from this extract, either from the germinating soybean seedlings or the lactobacillus acidophilus, you will take 1 ml of the enzyme extract. So, this can be reduced depending upon the availability. So, if you have less substrate available, so or the less enzyme extract, so you can also use 100 microliter. So, similar amount of the para-nitrophenol glucoside. So, this is 1 millimolar, it has been prepared for you in the phosphate buffer. So, this substrate and this enzyme extract you will mix and then leave it for incubation at 37 degrees centigrade for 30 minutes, and then you see how much product it has been formed, and then you can go for the calculation. And the activity, it is usually represented in terms of micromoles of the product formed per minute per mg protein. So, that means you should have idea about the amount of the protein present in the extract as well, right. So, this is you will be doing in the lab. So, if any queries, I would be happy to answer.
Sir, I wanted to ask, like, what is the applied usage of this? How we are utilizing this fat in some application?
No, as I said that the conjugated forms of the isoflavones, they have the less bioavailability, and we are dependent on our gut microbiota. But gut microbiota, it varies from person to person. It varies with the age. It varies whether you are under medication or not. If you are taking more of the antibiotics, so your gut microbiota, it will be poor, it will be destroyed. So, that means even if you are consuming the soybean, you won't be able to get the benefit of the isoflavones. So, that is why we are looking for this beta-glucosidase, which has the ability to hydrolyze the conjugated forms into the free forms. So, what we are planning to do is that we will use these probiotic bacteria in soya products.
Okay.
So, either we can directly add the bacteria, or we can isolate the beta-glucosidase enzyme, immobilize it, and then we can add in the product. So, all the conjugated forms of these isoflavones, they will be converted into the free form. So, that means now if we are consuming the soya product, we won't be dependent on our gut microbiota. So, already they are present in the free form, not in the conjugated form. So, this is the application of the beta-glucosidase.
Thanks, sir. It was a very nice explanation. Sir, I have a question that how to isolate the enzyme extract from soybean seedling?
Now, you are preparing the enzyme extract, you are not isolating the enzyme extract. From you are preparing the enzyme extract, you are isolating the enzyme.
Yes, sir.
So, there is a difference. Enzyme extract is you are preparing the enzyme extract and isolation of the beta-glucosidase. So, it is very simple process. Say we have taken 1 gram of this germinating soybean seedlings. So, usually we take 1:5 or 1:10 ratio of the buffer, and here the homogenization it will be done in the phosphate buffer. So, we have prepared the phosphate buffer for you. It is I think 0.1 molar pH 6.5. So, that is the buffer which has been prepared. So, using this buffer and the germinating soybean seedling in this ratio 1:5, so you can homogenize it and then do the centrifugation, take the supernatant, and that supernatant it will be used as the enzyme extract.
Thank you, sir, for nicely presenting the protocol in a brief way, and it was very simple and lucid. So now it's time to have a hands-on experience. So we'll go to the lab and we'll start the experiment. Thank you. Just give a big clap for the.
Oh, thank you, Ranjan. Yes.
Okay, welcome friends. Today we are going to do the beta-glucosidase enzyme assay from the soybean and two probiotic species, that is Lactobacillus acidophilus and Lactobacillus rhamnosus. So, this particular enzyme beta-glucosidase, it is very important when it comes to the bioavailability aspect of isoflavones. What these enzymes do is they convert the isoflavones into their more bioavailable forms, that is their glucosidic form to the aglyconic form. So, when in case of the enzyme assay from the soybean part, what we are going to do is we are going to make the enzymatic extract from the soybean leaves. Actually, the enzyme which is present in soybean is more specific towards the isoflavones. The reason being is because the isoflavones, they are naturally only present in the leguminous species. So, the enzyme it is more prone to show the specific activity towards the substrates. So, first we will prepare the enzyme extract from the soybean, and in case of probiotic species, what I have done is I have grown the probiotic species overnight in the MRS medium. MRS medium, actually this medium is specifically used for the culture of Lactobacillus and Lactococcus species. So, after growing it overnight, what I have done is I have taken the culture and pelleted down the cells, and then I have done the ultrasonication. So, in that case I have done 15 seconds pulses. I have given 15-second pulses for a total of 3 minutes for the both species, and then what we are going to do is we are going to prepare the supernatant from that also and supernatant from the soybean leaf also. So, here what we have is the soybean leaf sample. For the sample grinding, I am using the phosphate buffer with the pH of 6.0. So now we have prepared the extract, and we are going to spin it down at the 10,000 G for 10 minutes. And with this I have taken the 1136, which is Lactobacillus rhamnosus extract, and Lactobacillus acidophilus extract. So these numbers, they are JCM numbers which are assigned to particular strains on the Japanese collection of microorganisms. So 1132, that is used to identify the Lactobacillus acidophilus species.